Okay, good afternoon. Ladies and gentlemen, I would like to thank the Alliance for giving me the opportunity to talk today about MolMed and about some challenges in gene and cell therapy. Note that the presentation contains some forward-looking statements, so this by definition contains subject to risks and uncertainties. Now, in early 90s, first experimental treatments in gene therapy, namely ADA and TK, significantly contributed to the conception and birth of MolMed for gene and cell therapy services. And this actually paved the way for the growth and maturation of the company over the years into what is now an integrated biotech public company that actually is focused in developing biopharmaceuticals in oncology and genetic orphan diseases. We have now more than 150 employees, 75% are scientists. We have 425 granted patents, plus 60 under evaluation. We have 2 compounds in phase 3 clinical development and a conditional approval application file in Europe for Zalmoxis TK. MolMed has 2 technology platforms, including recombinant proteins and gene and cell therapy. NGR-hTNF is a recombinant protein that selectively binds to tumor vessels, not to normal vessels, and this allows its use at an optimal biological dose of 0.8 microgram per square meters. And NGR-hTNF has been extensively investigated in clinical trials, enrolling about 1,000 patients. Significant results have been obtained in large phase 2 randomized trials in non-small cell lung cancer, ovarian cancer, soft tissue sarcoma, and in 1 phase 3 randomized controlled trial in malignant pleural malignant mesothelioma, where the drug significantly increased the overall survival in patients with short treatment interval, treatment-free interval, namely those at worst prognosis. Now, gene and cell therapy is MolMed core competence, and promoting internal programs, including Zalmoxis for high-risk leukemia, and CD44 V6 targeted CAR T-cell therapy for liquid and solid tumors, and at the same time promoting GMP solutions focused on R&D manufacturing control for academic and industrial partners. In this context, we have a long-standing experience in genetically modified T cells, in hematopoietic stem cells. We have a proprietary product in phase 3, long-lasting collaborations with many charities, academia, pharma and biotech, and we promote a significant investment in quality management system and in control strategies, and in 2 state-of-the-art facilities for cell transduction and vector manufacturing with a total capacity of more than 30 clean rooms over a GMP surface of about 2,500 square meters. MolMed has 2 internal programs, as I said, in gene and cell therapy, and is an important partner of Telethon, TGET, and GSK in ADA, SCID, and for clinical programs exploiting the lentiviral vector platform. Now, there is one concept I consider of utmost importance, and this is that even the most striking scientific breakthrough needs translation to become clinically useful. And actually, to attain this goal, we need to face many challenges, and their complexity needs competence that actually comes from different players. And this includes patients and patient organizations to focus on unmet needs and critical clinical outcomes, academia for cutting-edge discovery and clinical development, biotech to develop processes and technologies, products, and industry to make this therapy available at large scale, along with regulatory authorities, to frame an efficient set of regulations to guarantee the safe access of this innovative therapy to patients. And thus, gene and cell therapy, in our opinion, holds promise also thanks to this integrated effort. And I would like to make briefly some example. The first example is TK gene therapy, Zalmoxis, originally devised by scientists at the San Raffaele Hospital in Milan and developed by MolMed. Acute leukemia patients undergoing haploidentical transplantations also receive TK transduced T cells, which overcome the need for immunosuppression because of the control of GVHD via the suicide gene system, and induce protection from leukemia relapse, protection from infections, and prompt abrogation of GVHD through the administration of aganciclovir, thus overall promoting rapid immune reconstitutions. Zalmoxis is the most largely test lymphocyte-based immunogene therapy of cancer, having enrolled more than 117 patients up to now. We receive orphan drug designation in both Europe and United States. We have patent protection up to 2030. Automation of manufacturing processes is ongoing, and the drug induces a rapid immune reconstitution associated with prolonged survival, regardless of the disease status at transplant. And interestingly, safety and efficacy data from Zalmoxis trials have been analyzed against data coming from both ABMT and CIBMTR registries. And the main conclusions include the fact that Zalmoxis halves the non-relapse mortality, particularly due to infections, and increases the overall survival. Graft versus host disease is readily controlled in all patients without the administration of immunosuppressive drugs. And this data will be presented at ABMT and ASCO next year. It's also worth noting that Zalmoxis gene therapy concept can be also exploited as a platform. And this is based upon the know-how in genetic manipulation of lymphocytes, the expertise and use of suicide gene system, and in the use of selection tools, which overall can promote the application of this know-how to apply this know-how to CAR-based cell therapies to target additional diseases and patients' populations. The second example is a successful gene therapy for ADA SCID. This was devised at the San Raffaele Hospital and was based on the gene correction of autologous stem cells. It was one of the central projects actually brought to the foundation of TGET in 1995. Over the years, MolMed has been involved in R&D, manufacturing, and control, contributing to making this therapy clinically available. As you know, GSK acquired the rights on ADA, signed with us an agreement to manufacture the drug, and last May announced the EU regulatory submission for gene therapy of ADA SCID. An additional example is given by two clinically successful lentiviral vector-based gene therapies, MLD and WAS, that are actually part of a broader spectrum of indications encompassing 6 genetic diseases. Thus, there are all instances of the fact that this integrated approach makes possible the translation of highly innovative therapeutic strategies to patients. In this context, we continuously promote the optimization of vector and cell product GMP manufacturing processes, scaling up both vector platforms, also in bioreactors, optimizing the downstream, attaining a highly increased removal of DNA of different origin, the successful introduction of double 0.2 micrometer sterilizing filtration, along with the cleaning validation for viral cross-contamination. Vector stability is long-standing in both platforms. We are introducing automation for transducing process, transduction process, And we also attain highly significant increase in transducing units and mean titer for vectors and high transduction efficiency along with the optimal vector copy number in the different target cells. Now, the process of translation from bench to bedside is difficult, lengthy, costly, and it has to profit and benefit of our learning and experience to face several challenges. And so in this frame, we believe that the development path with preclinical and clinical development should actually go hand in hand with the development of process manufacturing and control. And in this context, there are many challenges in process manufacturing and control in cell and gene therapy, including scale-up, robustness, and reproducibility of the process, the need for QC strategies to assess vector and drug products, analytical method robustness, but also the need to improve the method battery and the timing for readiness and release. There is the need to optimize the amount of vector and drug product needed for release and stability, and to develop new strategies to control the potential presence of genetic impurities in vectors. And overall, clearly make these approaches economically viable over time. Quality assurance is, in our opinion, another very critical aspect, including sterility assurance in closed systems, validation of aseptic process via media fill, and especially training and qualification of operators that risk to be a limiting factor, where maybe the automation of process and control might be a future reply. We need, therefore, for an evoluted and robust QMS, and also management of raw material and suppliers. We need continuous interaction with regulatory authorities to agree process characteristics and control. We learned about standardization issues and integrate increasing knowledge in the field. So, in conclusion, the biggest challenge is to translate science into clinical benefit for the patients. And albeit some very difficult historical moments, for instance, the FDA hold on clinical trials in gene therapy, or difficulty to raise capital. Albeit those, if now gene therapy is ready for prime time, and if Wall Street now is getting very interested and excited about these new avenues, is also, we may dare to say that it's also thanks to the contribution of determination and passion we, all of us, put into the field in the past 2 decades. Thank you very much for your attention.